This is Streptococci Bacteriology tutorial presented by Dassi SchrDbb. We reference Lippincott Illustrated Review of Microbiology by Cynthia Cornilesson, Bruce Fisher, and Richard Harvey. So here we're going to visualize Streptococci. So with Streptococci, you need to know that the Streptococci are in the group called free-living bacteria. We were also having another group called the intracellular bacteria. We have another group here in the free-living bacteria. All these groups were seen in the introduction of bacteriology. So you can just pause and then go and visualize those videos before you continue. Now we have the free-living bacteria is going to be divided into two, gram-positive and gram-negative. So we are not going to be involved with gram-negative. Gram-positive consists of cocci and rods. You are not involved with rods. The cocci are divided in Staphylococcus aureus. We have already seen what the cocci are divided into Staphylococcus. We have already seen a tutorial on Staphylococcus, so we are not going to speak about that anymore. The next cocci, we are going to have Streptococcus. Now we need to know that the Streptococcus and the Enterococcus are considered as Streptococcus, a different group of Streptococcus. Is it clear? So we are going to speak of both of them in this tutorial. Under Streptococcus, you have Streptococcus agalactiae, you have Streptococcus bovis, Streptococcus mutans, Streptococcus pneumoniae, Streptococcus pyogenes, and then the next group is going to be Enterococcus, still under Streptococcus. And under Enterococcus, you have Enterococcus faecium and Enterococcus faecalis. All Enterococcus and all the Streptococcus belong in a group called Streptococci. Is it clear? All of them are in a group called Streptococci. And then these are there are different groups of Streptococci that we're going to visualize later, which are going to have the different names. Now, let's visualize now the classification of the Streptococci. In the classification of the Streptococci, we have different class of Streptococci that we're going to visualize, as you can see here. So this is the classification scheme of Streptococci here. So for the classification scheme of Streptococci, you need to know that the Streptococci are divided into group A, Lancefield group A, B, C, D, T, even E, F, G, just continue to T and U. Is it clear? And then you have others. Is it clear? Now, the group A, we have Streptococcus pyogenes, and you see under Streptococcus pyogenes, you can see the letter alpha, beta, gamma, and others. The alpha just means that it is alpha hemolytic. Beta just means that it is beta hemolytic. Gamma just means that it is gamma hemolytic. It's just clear. Is it clear? So you have, if they put alpha or gamma, it means that it is alpha or gamma hemolytic. Is it clear? So we start here. We have A. This is Lancefield group A. Example of Lancefield group A. We have Streptococcus pyogenes beta, which is beta hemolytic. Now Lancefield group B. Example is Streptococcus agalactiae, which is beta hemolytic. Lancefield group C, we are not giving an example. Lancefield group D is Streptococcus bovis, which is alpha or gamma hemolytics. We have now Lancefield group even T, TU. We have other Lancefield, which are Streptococcus pneumoniae, which is alpha hemolytic, and Streptococcus mutans, which is alpha or gamma hemolytics. Usually we consider Streptococcus pneumoniae in group B Lancefield, is it clear? And even Streptococcus mutans is considered. So let's continue. We have the other here. In the others, you have Streptococcus pneumoniae and Streptococcus mutans, and as I've just said, Streptococcus pneumoniae is usually considered as a group B, a Lancefield group B Streptococci. Is it clear? Usually with mutants. Now we continue with the next one here. The next diagram here is going to show a schematic representation of the Staphylococcus, the Streptococci M protein. The Streptococci M protein actually is the antigenic protein of the Streptococci, and this M protein is what is going to result to rheumatic fever. Is it clear? This M protein is what is usually associated with rheumatic fever, which is associated with the group A Streptococci. When group A Streptococci, beta hemolytic Streptococci do an infection in the respiratory tract, Then later on you have a rheumatic fever, that is any form of rheumatic disease with different characteristics based on the Jones criteria. You are going to this is going to be due to these M proteins, because actually these M proteins resemble the antigens of the connective tissues. Is it clear? Since it resembles the antigens of connective tissues, the antibodies that are going to be produced against the M proteins of the Streptococci can cross react with the antigen, the normal antigens of the body. Is it clear? So this is the M proteins, this is the N terminus of the M protein, and this is the C terminus of the M protein. N just means there's a nitrogenous terminus of the M protein, and then the C just means there's the carbon portion of the M protein. And this is cell wall and the cell membrane. So all that is a schematic representation of that. Now the next thing is this is the virulence factor now of the Streptococci. Now we start with Streptococcus pyogenes. So Streptococcus pyogenes, it produces biogenic exotoxins. Is it clear? So there are certain exotoxins that are going to be produced that are going to cause the immune response to produce cytokines, and those cytokines are going to result to pyrogens. So there's a difference. There's what is going to be called pyrogenic, and there's another which is pyogenic. PYO is pyro, PYRO is pyro. When we say pyro, it means that it is producing heat. Pyrogenic means heat production, but PYO means post-production. So in this case, it is pyrogenic exotoxin. It's going to produce heat just because it's going to be the release of cytokines. But now this is Streptococcus pyogenes, meaning that it is these Streptococcus usually produce pus. Is it clear? But in this case, this pyrogenic exotoxin produces heat and not pus. So it's going to cause various effects, including rash, seen the scarlet fever. Scarlet fever is for Streptococcus, while the scalded skin syndrome is for Staphylococcus. And there's also a form of a toxic shock syndrome in case of Streptococcus. This is Streptococcal toxic shock syndrome. All these are pyrogenic exotoxin. But in the case of Staphylococcus, don't forget that you can just go and visualize that. And in those cases, they were super antigenic exotoxin that were produced in those cases. Now the next case of the next virulence factor of Streptococci is going to be the lysins. There is a streptolysin O and a streptolysin S. They are going to damage mammalian cell, resulting to the cell lysis and the release of lysosomal enzyme. So this enzyme is just going to be resembling as those toxins that was produced, the cytolytic toxins produced by Staphylococcus. So these are the streptolysin O and the streptolysin S, which are used in the lysis of the mammalian cells. Now we have another enzyme which is used here, which is the streptokinase. So streptokinase, unlike the coagulase that was in the case of Staphylococcus aureus, streptokinase here is going to catalyze the conversion of plasminogen to plasmin, which causes the lysis of the clot. Is it clear? So streptokinase is going to cause plasminogen converted to plasmin, because you need to know that there is what is called tissue plasminogen activator, because there is always plasminogen inside the blood, and the tissue plasminogen activator convert the plasminogen to produce plasmin, and plasmin is used in breaking down blood clots. Is it clear? So the streptokinase have that similar function in breaking down the blood clot, because it converts the plasminogen to plasmin. Is it clear? So causing the lysis of the clot and facilitating rapid spread of the organism. Is it clear? Because normally you need to know that one method to control the spread of organism is producing localized blood clot at a particular area where the organism is infecting. Is it clear? So streptokinase can break down this blood clot in order to make sure that the organism can be spread to a wider area. Now another virulence factor is the C5a peptidase. It's going to inactivate the complement factor C5a. Is it clear? Because you need to know that the complement factor is another innate immunity that is going to be in the body of all human beings. Is it clear? So in complement factor when it's activated is going to result to releases of the bacterial cell wall. Is it clear? But now with this C5 peptidase is going to inhibit the complement factor mechanism. Is it clear? So to prevent the production of the complement complex. Is it clear? That is going to inhibit now the releases of the bacterial cell wall. The next is going to be streptodonesis. Streptodonesis is a DNase that is going to degrade the viscous DNA in necrotizing tissue or exudate and aiding in the spread of infection. Is it clear? So actually the streptodonesis are going to degrade the DNA. So you're going to make sure that the DNA of the bacteria that have been destroyed are going to be splitted up. Is it clear? So when the DNA is going to be splitted up, it can easily be transmitted to different fluids that can be uptaken by different bacteria in order for them to know the method of resistance. Is it clear? So it's just like a way that there's a war between bacteria and there's a war. Just think that there's a war between bacteria and the immune. So just think that there's a war between the bacteria and the immune system, and then the bacteria is the enemy area. Is it clear? So the enemy army and the immune system is the barrier army. So let's think that you have one enemy. So in this case, the immune system is one army that is going to attack the bacteria. Is it clear? So when it attacks the bacteria, you are going to have the production of certain guns type that are going to be the DNA against those immune system. Is it clear? So to prevent the destruction of those guns, the streptodonesis are going to split the different gun types and going to make sure that other army that have not been yet have not faced against the immune response are going to receive those gun types, and then they can use those gun type in order to fight against the immune system. Is it clear? So that is the role of the streptodonesis. Now the next role is the hyaluronidase. So it's going to disrupt the organization of the ground substance, because you need to know that the ground substance of most of the connective tissues is made up of the hyaluronic acid. So in order to improve the spread of the bacteria, you have the enzyme called hyaluronidase that is going to destroy this hyaluronic acid for the spread of the Streptococci. Now this next image is going to show you necrotizing fasciitis. Now you need to know that necrotizing fasciitis is a polymicrobial disorder. Is it clear? But the major pathogen which is involved in necrotizing fasciitis is the Streptococcus pyogenes. So group A Streptococci. So this is how necrotizing fasciitis starts. So on day 0, we have a right lower leg edematous. So we have an edematous right lower leg with an edematous area below the knee. Then later at day 2, we have initial debridement. So we have necrotic tissues. The cells are going to die because these Streptococci are high they are phagocytizing on your normal tissue, causing lysis of the normal tissue, the streptolysin O and streptolysin S toxins and all that. So they are going to result to the lysis, and they are going to use their hyaluronidases and the streptokinase in order to spread from one area to another. So it's going to, in this case, you're going to have initial debridement reveal necrotic tissue underneath with a multiple layer of the thrombosed blood vessels. And then at the level of day 6, you have radical debridement was performed before the infectious process was progressed towards the knee. So this is the necrotizing fasciitis, one of a very very terrible disorder produced by Streptococci. Now, there are other disorders produced by the Streptococci. The first one, you have acute pharyngitis or pharyngotonsillitis, which is going to make clinically susceptible patients to rheumatic fever. The second is Impetigo. There is a Impetigo which is caused by Staphylococcus aureus. There is another form of Impetigo caused by Streptococcus pyogenes. The next is so the next is so after that form of Impetigo, you have now erysipelas. Erysipelas is usually a swelling of the, so there's a difference between erysipelas and cellulitis. So erysipelas is just swelling of the tissue. Is it clear? So you have erysipelas, you have a unilateral leg swelling, and then you have boundaries at the level of the swelling. So the swelling portion are going to be bounded, and usually is going to produce an erythema. Is it clear? So we have another one which is a puerperal sepsis. Puerperal period just means a period after birth. So when there is sepsis after birth, it is usually due to Streptococci. That's why these patients who that have given birth in aseptic condition, it's better to give the patients amoxicillin or any penicillins as a prophylactic. The next one, you have invasive group A Streptococcal disease. We have Streptococcal toxic shock syndrome. We have post-Streptococcal sequelae. Now, the post-Streptococcal sequelae include now acute rheumatic fever that was just talking about, and acute glomerulonephritis. These cases occur when there are autoantibodies that are produced that are cross-reacting with the tissue of the tissue cells. Is it clear? Why they were this cross reaction is because there is a confusion between the normal antigenic the antigens of the Streptococci, that is the M protein, and the antigens of the body, because the antigens of the Streptococci resemble that of the body. Is it clear? That those M proteins seen here. Now, the next one is acute glomerulonephritis is occurring usually after impetigo or pharyngitis, but this one is usually after an initiation of pharyngitis. Now, the next one is laboratory identification of Streptococci. Now, you have latex agglutination for identification of group A beta hemolytic. So you do, in positive you have this image, while in negative you have this image. Now, group B Streptococcal, so group B beta hemolytic Streptococci is now the next group. So this is, you need to know that for the case of Streptococcus pneumoniae, they are gram-positive, they are non-motile, they are encapsulated, and the lancet-shaped cocci. So this is how Streptococcus pneumoniae resemble. This is a picture of Streptococcus pneumoniae. It's an example of a group B Streptococci. So example, so as I've said, Streptococcus pneumoniae is a beta hemolytic. It means that it can also cause hemolysis of the blood. Now, you need to know that the Streptococcus pneumoniae, as I've said, they are gram-positive, they are non-motile, they are encapsulated, and they are lancet-shaped cocci. So they are usually diplococci, as you can see here. Now, now in this diagram now is going to show a comparison of the severity and the prevalence of pneumococcal infection in children in the United States. So you need to know that the least pneumococcal infection is otitis media. Is it clear? But the prevalence of otitis media is the highest. So every time a patient is complaining of any anytime you think of upper respiratory tract infection, that you have catarrh in a child or you have a child who is coughing, you must always examine the ear, because there is a high prevalence of otitis media caused by Streptococcus pneumoniae, but it is the least severe. Then after otitis media, we have the pneumonia. Pneumonia is the next prevalent, but it's also lesser severe. Now, more severe now is a bacteremia. Bacteremia, when we say there's a difference between bacteremia and septicemia. Bacteremia is just the presence of bacteria in blood, but the bacteria are not actively dividing. While septicemia is the presence of actively dividing microorganisms in blood. Is it clear? And the last one now is meningitis. Meningitis is when you have inflammation of the meninges, which is also the pneumococcal species. So this next diagram is going to show you the cytolytic toxin produced by Streptococcus pneumoniae. At first we were speaking about the Streptococcus pyogenes. Now we are speaking about Streptococcus pneumoniae. So the cytolytic action of Streptococcus pneumoniae is that you need to know that Streptococcus pneumoniae produce autolysins. So it's going to make that the Streptococcus pneumoniae is going to be lysed, and inside the Streptococcus pneumoniae you have now pneumolysin, and the pneumolysin is what is going to destroy the mammalian cell. I hope you have understood this part. If you have not understood, let's start back again. You need to know that Streptococcus pneumoniae have an endotoxin. That endotoxin is called the autolysin. It has an exotoxin, sorry. Streptococcus pneumoniae has an exotoxin called autolysin. So it releases that exotoxin, and that exotoxin is going to result to the destruction of the Streptococcus pneumoniae. Is it clear? Now the destruction of the Streptococcus pneumoniae is going to cause now the release of the endotoxin, the toxin that was found inside the Streptococcus pneumoniae. Is it clear? That toxin that is found inside the Streptococcus pneumoniae is now required to destroy the mammalian cell, and that toxin is called the pneumolysin, which is now going to result to the destruction of the mammalian cell membrane. Now, what is the age-specific rate of community-acquired pneumonia according to the different ages? So this is the different age-specific associated with pneumonia of different pathogens. So let's start with the first age group, that's 18 to 34 years, then 35 to 49 years, then different age group as you can see here. So the diagram in blue is due to Streptococcus pneumoniae. In yellow is going to be due to Mycoplasma pneumoniae. With red is going to be due to Legionella species, and with orange is going to be due to chlamydia. We see that at 18 to 34 years, all of them are the age-specific group. All of them are going to be more prevalent. These are the cases per 100,000. You see, the more you increase in age, the more you are prevalent to pneumonia. But all of them are usually associated with all the different pathogens at this age group. Now, between 35 to 50 years, you have more of Streptococcus pneumoniae and more of Mycoplasma pneumoniae being the cause of the pneumonia, and you have less of Chlamydia pneumoniae and Pneumophilia and Legionella pneumophilia as being the cause. Now, between 50 to 65 years now, you have another age group. See, there is more of Mycoplasma pneumonia, more Streptococcus pneumoniae, and there is less of Chlamydia pneumonia and Legionella pneumophilia. Now, between 65 to 75 years, the most prevalent is Streptococcus pneumoniae. Then after that is Mycoplasma pneumonia. Then we have Legionella species, and the least is Chlamydia pneumophilia. Is Chlamydia pneumonia. So this diagram now is going to show you laboratory tests that are useful in the identification of Streptococcus pneumoniae. So we have inhibition by optocin. So this is one laboratory test, inhibition by optocin. So we have growth of colonies of Streptococcus pneumoniae is going to be inhibited by optocin that is contained in a disc applied on blood agar. Is it clear? So you see that Streptococcus pneumoniae, or the Streptococcus pneumoniae is going to grow on the blood agar, but it's going to be inhibited by the optocin disc. That's one thing that can help you to identify Streptococcus pneumoniae. Another test to identify Streptococcus pneumoniae is a Quellung reaction. In Quellung reaction, the capsule of Streptococcus pneumoniae is going to swell in presence of specific pneumococcal antiserum. So you see on microscope, you are going to see that capsule is going to swell. You see big capsule, and then you see Streptococcus. So this is a positive Quellung test for you to swell the capsule, and this is a negative Quellung test. The next one is the lysis of the bile acid. Is it clear? So you need to know that bile acids such as sodium deoxycholate, so there are different bile salts and acids. Sodium deoxycholic acid is going to dissolve Streptococcus pneumoniae, and a clear turbidity is going to be dissolved by Streptococcus pneumoniae, and the clear turbidity is going to be seen. So here in bile acid negative is negative when the bile acid is not going to be dissolved. Is it clear? Why in case where it is going to be positive for Streptococcus pneumoniae, you are going to see bile acid is going to dissolve. So all that are tests in order to visualize Streptococcus pneumoniae. Now the next one is the incidence of vaccine type invasive pneumococcal disease before and after introduction of pneumococcal conjugate vaccine. So you see that before the introduction of pneumococcal conjugate vaccine, you see that the prevalence of the pneumococcal disease was very high. The incidence of pneumococcal disease was very high at less than 5 years before it was given. Is it clear? Now, after it was given, you see that the prevalence has reduced. Now, the next Streptococcus we're going to speak about is the Enterococcus. So Enterococci. So we start with Enterococcus faecalis here, which is going to showing a chain formation characteristic of Streptococci. So Streptococci, you see a chain formation is produced. So just Enterococci has that same characteristic with the chain formation. It can also be the Diplococci, but Enterococci is mostly a chain formation. Now, the next one is antimicrobial agents that are useful in treating infection with Enterococci. You need to know that what Enterococci, unlike other, have specific antimicrobial which are being used for them. So we have ampicillins, you have gentamicin should be used together in order to treat Enterococci. You must always add an aminoglycoside when you want to treat an infection caused by an Enterococci. Is it clear? So every time you think of an infection caused by an Enterococci, you must add always an aminoglycoside. An aminoglycoside is extremely useful when you think of an infection caused by Enterococci. Is it clear? Ampicillin is used generally for cocci, but Enterococci is highly resistant to ampicillin, so you must always add gentamicin when you want to treat an infection with Enterococci. So this is the first line. So the second line, if now these two fail, then you can use vancomycin, and you can use the quinapritin, dapropristin, or you use linezolid. Is it clear? Now this is streptococcal endocarditis showing vegetation of the mitral valve leaflet. So this is just an autopsy that has been done, and you can see the streptococcal endocarditis that has been shown here. Now you can just visualize a brief summary on the streptococcal diseases. So the streptococcal species, you need to know that there are different species of streptococci, but generally the characteristic of streptococci is that they are ovoid to spherical in shape, occurring in pairs or in chain. If they are in pairs, they are called diplococci. If they are in chain, they are called streptococci. Now, they are non-motile and they are catalase negative, unlike Staphylococci, which are catalase positive. In case of streptococci, they are more catalase negative. Most are aerotolerant anaerobes. Is it clear? So they are also anaerobes. In Staphylococci, they were facultative anaerobes. Here they are aerotolerant, meaning that they are anaerobes, but they also they are tolerant in oxygen, because they grow fermentatively even in presence of oxygen. In presence of oxygen, they can also cause fermentation. So we have culture on blood agar is another. So it's the best thing to culture them. So as you can see, you have alpha hemolytic streptococci can be seen here, and then here is a beta hemolytic streptococci on blood agar. On blood agar, alpha hemolytic streptococci, you see an orange color of hemolysis, while in beta hemolytic streptococci you see a whitish color of hemolysis. Is it clear? So an example of a gram-sing or Streptococcus pyogenes, you can see a streptococci chain, a chain structure of gram-positive. So there are different species of Streptococci that can be known. You have Streptococcus pyogenes, Streptococcus agalactiae, Streptococcus pneumoniae. It's a different species. So Streptococcus pyogenes is a group A Lancefield, and it is beta hemolytic, meaning that it's going to hemolyze the blood agar as such here. Streptococcus agalactiae is group B Lancefield and is also beta hemolytic as shown here, while Streptococcus pneumoniae is usually considered as a group B, though in this textbook it has been said to be in others, and it's also alpha hemolytic as shown here on blood agar. So let's see the different characteristics. In Streptococcus pyogenes, you have is going to cause acute pharyngitis or pharyngeal tonsillitis, it's going to cause acute rheumatic fever, it's going to cause erysipelas, it's going to cause puerperal sepsis, it's going to cause invasive group A streptococcal disease. Is it clear? So in order to treat this streptococcal pyogenes, you use penicillin G, you use clarithromycin, you use azithromycin. So as first line is penicillin G for streptococcus pyogenes. So you use penicillin G as first line. Then second line you can use clarithromycin. The second line we use clarithromycin, and then the next line we use azithromycin. So that's what you see here. In Streptococcus agalactiae, we have meningitis and septicemia in neonates. It can be seen there. We have because you need to know that Streptococcus agalactiae is one of the normal flora of the vagina. Is it clear? So when the mother is giving birth, it can easily pass through the nostril of the child or the gastrointestinal tract of the child, being spread to the meninges to cause meningitis and septicemia in neonates. It can also cause endometritis, still because it's a normal flora in the vagina. It can cause septicemia or pneumonia in individuals with impaired immune system and diabetic foot infection. So the first line is penicillin, then the second line is an aminoglycoside. Now, the Streptococcus pneumoniae in causing with alpha hemolytic, it can be acute bacterial pneumonia. It can cause otitis media and can cause meningitis. The first line is penicillin G, or still as first line you can use cefotaxime. Cefotaxime is a second line a second generation cephalosporin, and you can also use ceftriaxone, which is a third generation cephalosporin, and you use vancomycin in second line. Is it clear? So this is an example of a fascial erysipelas. Is it clear? Fascial erysipelas. Now the difference between a fascia and a erysipelas and a cellulitis on the face is going to be a sign, the Millian sign at the level of the ear, where a cellulitis is not going to involve the ear lobule, and then the erysipelas is going to involve the ear lobule with Millian sign. This is an example of Impetigo, and this is an example of streptococcal pyogenes pharyngitis. So from here we are finished with streptococcal. You can say thanks for your kind attention. Don't forget to like and subscribe for our channel. Thanks.